Okay, hi Balticon, and welcome to Crossing the Boundary from Mostly Dead to Alive and Back. I'm your moderator, John Ashmead, and I propose the subject of this panel on a cue from John Skylar, who is one of our distinguished panelists. The description is, how is it something like the coronavirus can be completely inert one moment and then spawning millions of copies of itself the next? How did obligate parasites, organisms that can't survive without a host, manage to come into existence? What of transposons, jumping genes, viroids, and the dreaded giant virus? There are a host of things that seem to be dead one moment and then alive the next. How do they manage to navigate that curious gap from life to death to life, spores and all sorts of things? So it's a fun topic. And I'd like to start, as is traditional, with our various panelists introducing themselves. I'll start with John Skylar. Take it away, John. Brief description.
Thanks, John. Hi, I'm Dr. John Skylar. My background training in biomedical sciences focused on emerging RNA viruses, specifically those entering humans from bats, but expanded to fit into paramyxoviruses more generally, and the virus-host interactions that lead to immune responses and whatnot. I'm currently an associate medical director in the pharmaceutical industry, working on a non-viral topic, actually, which allows me a little freedom to talk about viruses publicly. And in addition to that, I am a writer of fiction and nonfiction, though my chief active project right now is pandemic-related, a newsletter called COVID Transmissions, which you can find on Substack. I'll put a link in the chat because I really should.
Okay, thank you. James?
I'll actually say I read John's COVID Transmissions every day, and it's great. Good news to keep up with everything. So my name is Dr. Jim Prago. I am a naturopathic doctor by training. I used to have a private practice. I discovered I love medicine and I hate business. So I also have some background in emergency medicine. But for the last, well, some of it overlapped, but for the last 15, 16 years, I've been a adjunct professor of biology at Malloy College, LIU, and Nassau Community College out here on Long Island. And so a lot of this, I'm coming from more the side of viruses and other things that can infect human organisms. And, microbiology, of course, is one of the courses I teach. So that's where I'm coming from.
Excellent. Doug?
Hi, everyone. Thanks for having me and inviting me as a panelist. My name's Doug Deluzin. I have a doctorate in genetics, human genetics that I got at. I study age-related diseases and health disparities. Until recently, I was a faculty at Morgan State University studying how the stress of the environment influences gene expression in our immune system. But I have training in infectious disease and how viral infections can play a role in our downstream health. And now I write about science at the NIH. So thanks for having me aboard.
Okay, great. Pam?
Hi, I'm Pam Garrettson. I'm a doctorate in wildlife biology from Louisiana State University, and I did my undergrad in biochemistry here at locally at UMBC. So right now I'm a wildlife biologist with the U.S. Fish and Wildlife Service. I'm not a virologist. We do deal with viruses, obviously, in particular avian influenza and the fallout that can come from that. And so that's kind of the direction my interest comes from, and I'm looking forward to learning from all these folks.
As am I. Anna?
Hi, so my name is Annakashina. I'm a professor of biochemistry at the University of Pennsylvania, and I have a virology background. My undergraduate degree in virology, and currently we're working on coronavirus and trying to identify post-translational signatures on coronavirus disease. So,
wow,
yeah.
So front lines.
Yeah.
Okay, well, I got the idea for this panel from John's blog. He expressed he got into virology because of his initial fascination with the transition from alive to dead of viruses, and to reward him or punish him for this insight, I'd like to ask him to start with that subject and just take it away. And we'll just all play into that and see how the conversation goes.
All right, well, I'm happy to kick it off in that respect, but I do think that my views on this are a little bit unorthodox. I think that there are a lot of folks out there that insist that viruses are not alive. I actually think that they are kind of a two-state mechanism. For me, the definition of life represents almost anything that can be described as a sort of chemical ship of Theseus. So a chemical process that is self-sustaining to where the original components that started the equilibrium are gradually replaced by the processes themselves, and that propagates it over time to where they've all been replaced, but the process continues going. And that can describe anything from your most basic self-replicating molecule to something as complicated as the human species. When it comes to viruses, I think that we get confused between the concept of the virus itself versus the dynamic chemical process that is the virus infection. And so I think of the particle, the virion, the virus particle, as not alive because it is similar to a seed, like a sesame seed. It's not alive until it germinates within a cell in its own way and begins the process of subverting that cell to its own chemical process. And I probably said process enough. The outcome of all of that is that the cell's life becomes the virus's life. And that process of the virus and host interacting in a way that either the virus subverts the cell or the cell beats back the virus in some way is where I've spent my intellectual curiosity for most of my career.
Wow, it sounds like a meme almost, jumping from one substrate to another and moving itself on like some kind of malicious tweet. Follow-ons from anyone?
Well, I used to be very adamant that viruses were not alive. And partly because of John's conversations with John, I've kind of changed my idea on that a little bit, because there are other obligate intracellular parasites that I do consider alive, like certain types of bacteria. But still, I still kind of consider viruses the zombies of the living world, because they're not really alive. They don't do all the living stuff that you usually expect. But what do? They come in and they eat up the brains of a cell and take everything over. And now that cell is churning out copies of it. So there you go. Viruses are zombies.
Anna, you look like you have something to say about that.
Well, so I had my training from the Moscow University in virology, actually. And we were taught that viruses are the simplest life form. So it's, so it can replicate, it has nucleic acid. And so different viruses take different modes of replication. So some viruses integrate into the genome, other viruses just use a cell as a host. But to me, viruses are totally a life form. And it's kind of,
I was amazed to hear of viroids, just little tiny circles of virus stuff, smaller even than an actual virus. Is a viroid the smallest thing that can somehow manage to get itself replicated across generations?
That's such a tough question.
Maybe a prion?
Yeah, that was what I was gonna say, Jim. I do think that we should lay out though what a viroid is.
Okay, yes, if you would do that.
So a viroid is actually a naked nucleic acid, an RNA molecule that can live in soil, and that is capable of infecting plants. Specifically, all of them infect angiosperms, and they are self-replicating RNAs. It's not, I don't know if it's as simple as it can get, but it is about as simple as it can get. That having been said, I'm not the only one that should talk.
Yeah, that ties into the hypothesis I've heard of RNA World, the notion that the whole cycle got started from RNA rather than from the more complicated DNA plus proteins. Doug, you want to pick up on that one? I see you nodding.
Well, I spend a lot of time studying RNA expression and particularly in humans. And I think that I think there's some interesting evidence against the RNA World Hypothesis, though I'm still firmly in the camp of it. For a lot of the reasons is that if you look at what RNA does in the human body, not only is it the messenger for protein coding genes, but a lot of RNA, most RNAs actually just function on their own. As I think that the central dogma is kind of unfortunately taken away their predominant role. They signal in like hormones, for example, between cells. They get packaged up and get transmitted from different cells and different tissues along the body using the bloodstream and the lymphatic system as kind of the highways. They can free float in our and avoid degradation because they're so small, like these viroids, and they can actually attach to cells and trigger signaling cascades from the outside of the cell to the inside of the cell and induce a genetic response, induce an inflammatory response. I think that a molecule that has such a broad depth of scope when it comes to self-replication, when it comes to signaling, when it comes to the ability to avoid it being degraded, I think you got a lot of bases covered when it comes to just starting out in the world. So,
yeah, and one of the things that amazes me about viruses is that, you know, that old expression, you have to keep your enemies close and your hosts closer, I guess. That if the virus ever lost its communication with a host, it might have extraordinary difficulties reestablishing its parasite-host relationship. So if there's a single break in the chain across the generations, it might be toast rather than hosted.
Yeah.
Oh, there was a question, two questions. One question about what a prion is. So I should mention.
Yes.
So there's kind of call them good prions and bad prions, I guess, because the actual what the prion molecule is supposed to be is a neuroprotective protein. But however, sometimes it can become misfolded. And usually what you can do with proteins is you change the pH, you increase the temperature, you use an enzyme, you can break down a protein. These misfolded prions, they're highly resistant to these kind of things, more resistant than microorganisms usually to those kind of things. And what they do is when they interact with a normally folded prion, they cause that prion to now misfold and become the quote unquote bad prion. And you get this buildup of these bad prions in neural tissue, and you end up with a type of what's called a spongiform encephalitis, because it makes your central nervous system, like your brain, look like a sponge. And this is mad cow disease is an example of this. And in humans, I always pronounce it wrong, but I can't even pronounce it.
Creutzfeldt-Jakob disease, I think.
Yes, thank you.
Yeah. So I wanted to add, because I work on prion diseases right now.
Oh,
so,
nice.
Just to clarify, so prion proteins exist in normal cells. They are like completely normal, and even though their function is not completely understood, I mean, we know a lot of things that they do, so they're essential for human bodies. So what happens in prion diseases that the prions get misfolded, and this misfolded conformation is supposed to be, so if you have one misfolded prion, it's going to cause other prions to misfold. So it's kind of, this conformation is malicious and it propagates. So when I was an undergraduate student, which was some years ago, like many years ago, actually, we were taught that prion diseases are caused by protein-based infectious agents. So at that time, people believed that this is the life form based entirely on protein. So we know now that this is not the case. This is not a normal protein being misfolded, but prion diseases, they're kind of a class of diseases which are infectious, even though they're caused by normal proteins, just because other proteins propagate. And mad cow disease is one example that we know best, but every species pretty much has its own prion disease. Creutzfeldt-Jakob is the one in humans, as well as Kuru, and they can also pass from species to species, because this abnormal conformation of prion proteins is very contagious.
Could I mention just a fairly important one in the North American wildlife world too, is chronic wasting disease.
Yeah.
In deer, and it started, I believe, in Wisconsin, and it spread through, I don't know, more than 30 states. And, there's deer are a very popular game species. So there is this concern, there's surveillance for chronic wasting disease, asking hunters to bring their carcasses in for testing, and just basically advising people, definitely when they're butchering these animals, avoiding any spinal cord or brain tissue if possible. There are certain butchers who have like the ability to butcher things very, very carefully that way. So that's sort of a real world potential issue. And I don't know that because it takes so long for these diseases to show up. I don't know that we don't have evidence of it yet, but do you want to take the chance of it in humans rather? Do you want to take that kind of a chance?
Yeah. Yeah, actually, so one of my projects in my lab is on chronic wasting disease right
Oh, no
now.
kidding.
And yeah, so it was identified for the first time pretty recently, but it existed for a long time.
Mm-hmm.
And it's kind of, I think people who study those diseases, they don't talk about the fact that this is essentially all the same disease. If you talk about chronic wasting disease or mad cow disease or any others, so they basically can pass to each other just through protein conformation. And I think, people kind of don't realize that. So yeah, it's a big problem.
Right. No, I mean,
Yeah.
I am not, I've changed, I would say, my behavior around
Mhm.
how willing I would be to eat somebody's venison, depending
Yeah,
on.
yeah, I'm the same way here.
Yeah, it was
For my edification, actually, can I ask? So my understanding of this, and I want to make sure I'm not outdated. My understanding of this is that prions can emerge both in a communicable way as well as spontaneously within a single organism due to misfolding. Is that right?
Yeah, that's correct. So prion protein exists in every species, and occasionally it misfolds, and apparently this misfolded conformation is so favorable that it forces other prion protein in the same body to misfold. And I think because of this, it's very easily transmitted to other species, and people tend to go by like what examples of transmission they have seen, but in reality, like from a biochemist point of view, there's no barriers to species from this. So just
That's awesome that getting to the notion of life as a kind of process or pattern, which I really like. Prions might be therefore almost kind of the minimal self-reinforcing pattern.
Mhm.
I just have this terrible picture of a shelf full of origami going bad. They're all folding the wrong way. One
Yeah.
crane after another misfolding itself.
right.
It's almost like the Sorcerer's Apprentice.
Mm.
You know, it's this magic that gets out of hand, and no matter how much you try to cut it in half, it just keeps coming back and subverting more and more to its will, not to anthropomorphize too much.
Yeah, we can. We're free to anthropomorphize. I don't think the prions and the viruses have lawyers yet.
Hopefully not. We don't want to.
Yeah, I've been doing a talk on AI, and I cover the risks of AI. And one risk of AI is known as the paperclip catastrophe.
Mhm.
You have this civilization which is more and more reliant on artificial intelligence, and you've told the artificial intelligence to optimize, and for some reason you told it to optimize production of paper clips, and it promptly turns your entire civilization into raw material for generating piles of paper clips.
Yeah.
And there's a little bit of that to some of these viruses and so forth. Once they get going, once they figure out, and we can't help anthropomorphizing. We say once they figure out,
Mhm.
they're just randomly trying stuff, and it feels like intelligence to us because we anthropomorphize our opponents.
Well, you saying that it sees it as raw material, that's kind of the definite, like every single living thing is like that. And what makes it not happen, turning the entire world, is the fact that there's millions of species interacting with each other in ecosystems that kind of make it put things in balance. So it's all this grand balancing act, and I always, when I talk to my students, I always use the term Goldilocks zone so often. So when things are in the Goldilocks zone, everybody's happy. If you leave that Goldilocks zone, then things can get out of control and you can end up with a planet of paper clips.
You've reminded me of a horrible scene in The Matrix where Agent Smith says that humans are a virus, and he uses this analogy that humans will consume all of the resources available in a given area, unlike other animals, and we're like a virus because we will consume those resources without regard to the damage that we're doing. And I think that this is really unfair to viruses. I don't contest that humans will consume resources without regard to the consequences, but I actually think that viruses are chemically evolved to really be sensitive to resource limitations. And there are many viruses, such as the one that causes chickenpox, that will behave themselves very latently within a human body for decades upon decades upon decades until favorable conditions emerge, replicate themselves without causing lasting damage, and then proceed to re-enter dormancy without consuming all of the host resources. And that is all pre-programmed genetically by evolution, of course. But if anything, I'm saying this to combat the view that viruses are sort of unrestrained killers. They're actually quite sophisticated in their balance with the host.
Yeah, because if you kill your host, you're out of luck.
Yeah. And I think this kind of touches on like this discussion of whether coronavirus is a biological weapon. So biological weapons are designed to just kill everybody, kill your enemy. So coronavirus is actually this design very sophisticated because it kills about 1% of population, and it probably will evolve to kill less. So the virus wants to replicate, it doesn't want to kill its host. So the less host it kills, the more successful the virus is. So that's kind of,
Wow.
Just to me that the language that even our expert Anna here uses, though, it doesn't want to kill its host. That want, like it had, like we still thinking of it as something that wants to do something or not do something.
Right.
And I would also just add, like, yes, humans will consume all the resources available, but so will a lot of like different species. It's just that there's really not a whole lot to keep us in check anymore.
Our resource consumptions. I mean, climate change, that crisis is a direct side effect of people who didn't maliciously plan to overheat the planet. They're acting on rational economic imperatives. And for a long time, we're not even aware that we were having this side effect. People don't say enough good things about nuclear war. And one of the positive side effects of nuclear war was our understanding of climate change. That the initial studies of possible human impacts on the world climate were a result of worrying about nuclear winters. And it was realized that we could have global scale impacts on the planet. And so that we, as a somewhat unchecked virus, have to think a little bit more about what we're doing to our host planet. That we are responsible for that. I digress slightly, I apologize, but.
Well, if I could chime in for a second, I think it might have been James, you had mentioned the Goldilocks kind of idea, and that if you look at coronavirus, it's, and I would say similar like flu-like viruses too, are really perfect viruses for a pandemic where the incubation period varies, the infectivity and the ability to spread varies too. The transmission of it is kind of variable. If you look at something like Ebola, for instance, that is also an extremely deadly virus, but it's almost a virus that's too efficient at churning through. Someone gets affected with Ebola, they could be deceased a day later. And that actually limits the capability of the virus. If the virus were a thinking thing, it limits its ability to transfer to a new host, and it needs the new host. So, an Ebola pandemic would be horrible. It may also never happen because that virus is just too fast. It just doesn't have enough people to seed itself. And so I think it's something when it comes to evolution, I think the variations in the genes and certainly how it interacts with the host is going to play a role in some of these longer, more durable. Certainly any viruses that sit in our genome for decades are going to be viruses that'll probably stick around for a long time. Herpes, a lot of the herpes viruses do this, HPV, which causes cancer too. Those are ones that they sit around for a long time, and it's a lot easier for them to spread when you don't even know that they're there.
Yeah, and also a lot of it has to do with the symptomology. One of the reasons that polio, for example, is able to be so disseminated among human populations is that it doesn't cause symptoms at all in most people. As you mentioned, similarly, the coronavirus has the option of asymptomatic impacts, or at least not noticeable symptoms, more like gastrointestinal symptoms. Whereas Ebola virus, you really start to bleed from all kinds of places, and it's incredibly debilitating. And one of the things that is essential for virus propagation in human populations is movement. We don't spread it if we don't move. And even if Ebola was not so deadly, if it caused you to be unable to move during, well, and as it does, during the infectious period, then it is much less likely to spread widely. So there's a lot of almost behavioral elements to these things. And it really, viruses are incentivized to survive by what the disease they cause allows them to do to survive. To get away from this language of wanting, they have to find a solution to a biochemical problem, and we speak often of what that solution is as what they want, but really it's just they do what's allowed, and they do everything that's allowed, and they move forward in that way.
Yeah, the fact that quarantine is traditionally very successful against I don't know if it's a majority, but certainly a lot of diseases, is because the plague, which was the original motivation for the quarantine period in medieval Europe, will express itself fairly quickly, so that if you go 40 days without symptoms, you don't have the plague. I remember a delightful episode of Fireside Theater where the contestants in their game show, which was Name That Disease, were required to name the disease they'd been infected with before it actually killed them.
Oh, that's brutal.
And then at the end of this, the guy is desperately trying guess after guess after guess, and the announcer goes, "I'm sorry, you've got the plague."
The quarantine thing. One example of how well it works, we quarantine and face mask and distance and limited touch, although touch now we know is not that big of a thing with coronavirus, but we did this with coronavirus. Look at this recent flu season. I think we've had less total cases in the US than we had deaths the year before. So quarantine works.
One quick comment I'll mention too. I had brought up Ebola, and something I was just thinking about when you mentioned zombies too, is some great buzzwords flying around here. Is Ebola too is it's one of those where those individuals can die very quickly, but it also can hang around in the body for a long time. So even if it goes through a community, when people go in after, maybe a month after, and pick up the bodies to cremate them or bury them or do whatever's within their religious behaviors, that can still spread. Contact with just bodily fluid that's contaminated can kind of bring the zombie back to life, so to speak, and start a whole new wave of infection. So proper PPE and that's all that's going to be really important, and certainly wouldn't be helpful 200 years ago if people were battling this, because there wouldn't have been a lot of that proper PPE. So
I was just gonna say, I know there were some societies that ended up having to actually change their funeral rituals around the Ebola issue.
Yeah, although actually I'll mention during the plague, medieval people got quite wise to the idea that interacting with bodies of the deceased from the disease could actually spread it further. There's a code of rules from the city of Milan that describes how the people that actually cleared out the dead from plague houses were to be selected, and how they were to be interacted with by the rest of the people, which is to say they weren't supposed to be. And it was a job that you only had to do for a certain period of time, and if you were lucky enough to survive doing the job for that period of time, then you could move on and then not.
One of the things
Retirement.
that puzzles me about this whole dead to alive to dead transition regime of, the subject of this panel is how something which is basically sitting there knows when it's supposed to get going again, because it's like inert, and yet it gets ert again, if that's a phrase, or out ert. So you've got things like, of course, the coronavirus, you have spores in general, you have everybody's favorite microscopic teddy bear, the tardigrade, and you just have a long list of things that sit there for extended periods of time. It's been suggested that, say, microscopic life from Mars might have actually seeded this planet, hanging out for 2 million years on a meteorite kicked off the Martian surface, and then descending here to start things, a variation of panspermia, not apparently completely ridiculous. So how do these things manage to go from inert to ert? Comments?
Well, I think so. One thing to remember is that these things don't actually have any intelligence, the way we understand it. So they look at favorable conditions. So if they have like favorable hosts, like favorable conditions to propagate both inside the host and to other hosts in the population, they would prosper. If not, they will just die out. So my sense is that they actually kind of try out periodically, and whenever the conditions are favorable, they just go for it. So with coronavirus, I think one of the theories of its propagation is that it actually existed in the environment for a long time, but it kind of came to the point when the conditions were favorable for it to propagate, and it obviously did very well this time. So for a virus, it's pretty successful, even though I think if it causes less death, it would be even more successful, because then people won't isolate. So flu virus is more successful than coronavirus if you discount the 1918 Spanish flu. So I think, yeah, just like the evolution is believed to be like a process of trial and error, even though there are some contesting ideas to that, I think the viruses and the pathogens are the same way. They go through trial and error, and then when the conditions are favorable, they just erupt, basically.
A lot of it is just basically it's the concept of mostly protein. Sometimes it can be RNA, but form defines function, right? So you have these, like on the coronavirus, you got spikes, and most viruses work this way too. You have these little protein things sticking out, and when they bump into something that they can interact with, in other words, a receptor on the host that's usually used for something else, but the viral spike can bind with it, that causes a change in configuration of the protein, which then causes a change in configuration in another protein and another protein and another, and that is basically you've turned on the mechanism now of then the virus binds with the host, and then the DNA genetic information gets injected, etc. It's all just about, hey, something tripped a change in form of a protein, which then now makes it do something or stop doing something, and that makes another, and you just get this cascade of effects. And that's actually how receptors work in general, like hormones, right? You have a hormone receptor, hormone binds with the receptor, that causes a change in configuration of the receptor, which turns on an enzyme, which changes ATP into CAMP. CAMP turns on another protein, which phosphorylates another protein. Boom, 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 boom. Now with one hormone binding to one receptor, you have a million proteins doing something different in your cell. So, basically the virus kind of hijacks that kind of receptor mechanism for its own purposes. And just because the viruses that happen to be able to bind to a receptor are the ones that could continue with their mechanisms, and the ones that can't, they can't. Like a lot of viruses don't have a lot of error checking mechanisms, for example. There's a decent percentage of viral particles that are created from a cell when the cells burst open and all those viral particles come out. Many of them aren't functional because they're different in structure and they can't interact properly.
Yeah, I think it's important that what you've said there about the elaborateness of the program that these things engage in, because if we look at SARS-CoV-2, we've exploited actually its elaborate program in order to create the mRNA vaccines. The spike protein of SARS-CoV-2 is highly processed in the actual life cycle of the virus. It has different points where it is cleaved. It has different points where it actually changes shape. And so the moment that it switches from, well, sort of inert particle to infectious particle occurs when the spike protein interacts with the ACE2 receptor. And the protein undergoes a change from what's called the pre-fusion conformation to the post-fusion the fusion conformation. And that is actually a change in the shape of the protein that allows it facilitates its ability to actually fuse with the cell, either within an endosome or at the surface. And what has been done in the vaccines, and it was done in January of 2020, because we understand well these spike proteins, is a modification of that spike protein so that it cannot enter the fusion conformation and it remains stuck in the pre-fusion conformation. But that moment that John is asking about, we've actually exploited the machinery of it to create something that is more inert and more amenable to use as a vaccine candidate than it is as a virus protein.
Well, that's a fascinating description, James and John, that it's basically a kind of kiss from the target cell, which wakes up the coronavirus, which was inert until the cell did something to get it going. And I've been thinking, in terms of spores, where they're in an environment, say with water or whatever, which, causes them to come to life, but this is another mechanism for doing that. So that was a really interesting answer.
Another mechanism too is temperature. So you mentioned how could we have, say, panspermia, say life began on Mars, for instance, and traveled here. Cold storage, you're in the deep of space and it's so cold that particles almost don't even move. That's why, in the lab, we can freeze cells in liquid nitrogen, and 2 years later, you can thaw them and you can have liver cells or even brain cells growing in the culture again. It's just, whether or not the cells have the structural integrity to survive that rigid framework that they're going to be in for that long. But viruses and small bacteria, they're so compact and so small, without having to expel any energy, because you can't move anything. You're just kind of stuck in time until it warms up a little bit. It hits the ground, it warms up, it interacts with water, and now they're free just to kind of continue to do what they're doing. I think thermodynamics and temperature, energy usage is a huge player when it comes to the capability of some of these things to come out of permafrost after being frozen for so long.
Yeah, I think so. It's important to kind of point out, so viruses are different from like, for example, plague, because they are subcellular organisms. They have to hijack a cell to replicate, unlike bacteria, which are just kind of cells in the cells, and they can replicate without the help of the host cell. So I think, a lot of things that were said here kind of need to make this distinction of, and coronavirus, yes, it's one of the mechanisms. There are many mechanisms viruses use to replicate. So one of them is coronavirus, but we have many others. Right?
Yes, my prep for this panel was full of amazing discoveries. I read an account of an adolescent T-Rex, which was unexpectedly stuck in a mudslide and fossilized almost intact. And the Jack Horner and his fellow fossil hunters dug it out. Part of it, cracked open as it was being moved around. And they handed a few fragments of this to a biologist to see what she could find with it. She inadvertently left it, too long in the acid bath or whatever, and distilled it down to a bunch of collagen proteins, a bunch of collagen. And it turned out that the stuff was still biologically active. It could be cleaved by some kind of collagen cleaver. That was, they're able to actually see the structure of the collagen be preserved from this adolescent T-Rex. And it was still reacting with the appropriate enzymes. Not quite up to Jurassic Park levels of identity preservation, but still awesome, 65 million years. So that was another, there were a couple of other things. One of the things we haven't really talked about here, we've been talking about things that are normally recognized as organisms, or of course, prions are just like little bits of bad fold. But was a wonderful mention of transposons, bits of genes acting on their own behalf, which would be a subject we haven't touched on yet. Anybody want to talk about bad genes or jumping genes and stuff like that?
Well, there's actually a theory that viruses actually came about from something called retrotransposons. Because retrotransposons, these are sequences of DNA. There's some DNA stuff in the middle, and on either side is kind of the transposons. And what can happen is it can be transcribed into RNA, and then it can reverse transcribe itself back into DNA and then be inserted somewhere else. Now, one of these theories of how viruses evolved was these retrotransposons ended up being able to move from cell to cell. This is one of the theories of how viruses first developed. Regular transposons, they're more a cut and paste. What happens is it encodes for an enzyme. The enzyme cuts either sides of the transposon, and then it gets pasted somewhere. Sometimes it's a copy paste, sometimes it's a cut and paste.
So we can have some kind of horizontal gene transfer even among people. I know when I was growing up, each species was basically stuck with its genome and it couldn't do any sharing. And then I found that bacteria love sharing their genes. And apparently there are possibilities of even very complicated large organisms like ourselves doing a bit of gene sharing.
A human placenta. Oh, sorry, go ahead.
I was just going to say, what do you get? Bacteria just find random bits of DNA on the street and take it up.
Yeah, the human placenta is I think it would have been impossible for us to have evolved without the help of viral integration into the ancestral genomes of our ancestors. Some of the genes are influential in allowing our bodies to create that.
Yeah. So I just wanted to kind of point out, so yeah, we cannot say much about evolution because we weren't there when it happened, but I think viruses are believed to be one of the primitive life forms that I think evolved before the cellular organisms. So I think it's more like retrotransposons are believed to be traces of viruses. And there are some viruses that integrate into the genome. Other viruses never integrate into the genome. They just act like true parasites. They enter the cell, they replicate, and they leave. But yeah, retrotransposons is another very interesting example of how genes can transfer horizontally. And even though we don't know the origins of it, this is certainly a way to exchange genetic material in different ways. So.
Yeah, I think Anna points out something that is missing from the biology curricula that we have widely, particularly in the US, that there's a view that life proceeded to cellular life first. And it's not clear any longer that that's true. It may well be that there was a world of enzyme processes that were not constricted to these membrane-bound cells as we think of them now. It's possible that the viroids that we mentioned earlier are actually an artifact of a world like that. At the very least, they demonstrate that it is somewhat possible for an really unprotected nucleic acid to exist in a hostile world and propagate itself. So, and I think Doug made references earlier, the RNA World Hypothesis, that we really may have had a very chaotic primordial soup without cells in it, and some of these things may have emerged there, and eventually cells came about.
Actually, I'm surprised you said this, but yeah, that's very kind of a deep comment, because obviously I was educated in another country, and in my education it was obvious, yeah, that protein forms evolved before nucleic acids, and RNA evolved before DNA, and yeah, viruses evolved before cells. But yeah, it's all of us can only guess how that actually happens, but there are experiments that show that this makes perfect sense, at least.
Yeah, well, I think in my undergrad, even in which was in the 90s, I think it was mentioned in some of my classes as a kind of a new idea. But RNA might have been around first, and I remember in the class discussing, well, you got ribosomes that have RNA that do enzymatic stuff, and you have these other RNA that do these other things, and so so it gets mentioned sometimes.
Yeah.
Yeah, and I'm just amazed over the course of my life how much more complicated our picture of life has gotten. It was fairly simple and standardized when we thought that some amino acids had been hit by lightning and sort of got their act together and worked in a coordinated way. And then we started to see I think the part of the expansion of our imagination came with the black smokers at the base of the ocean, that volcanic vents with a surround of tube worms and all sorts of organism living not on life ultimately, but on chemical reactions, said, well, maybe there could be all sorts of strange ways for life to get started. And I think that's accounted for some of our enthusiasm for looking for life on Mars and the moon Enceladus and so forth, that all life here seems to have come from a single point of origin. Or at least there was one winner very early on and the other guys just got turfed. No way to tell that now. And there might have been very different forms of life, just by chance starting in one direction or another, and all this strange death to life stuff is giving us some kind of insight as to what the alternatives are. Would anybody care to speculate on some form of life which is plausible, some transition from not living to living, which we just haven't seen on this planet?
Well, I have one that I'm fond of.
Okay.
I think that there's a lot of electronic information in biological systems that we don't think about. DNA is a charge carrier, RNA is a charge carrier. We have these large networked molecules, and it would surprise me, let's say, but it would not be beyond the realm of possibility that there could be patterns of electron flow through these molecules that themselves are sort of propagated and that may represent some kind of self-propagating pattern. And could perhaps be communicable across cells or communicable by transfer of DNA. And those could be somehow impacting life and interacting with them in their own way. And then we would have a big question as to whether or not a pattern of electron flow constitutes life or not. But it's something I think about sometimes.
I saw a discussion of chlorophyll and the mechanism that it uses for capturing photons, where it relies on quantum level effects, to secure the maximum amount of energy from photons. I know there's a lot of stuff out now about quantum effects in biology. Most of biology is really classical in its treatment of chemistry. But there could be very subtle patterns within, say, neural response to incoming signals and so forth.
Yeah.
That's a wonderful idea. Subtle patterns of, say, surface electronics, which we simply haven't thought to look for.
Yeah, I think so. As a peer reviewer of scientific papers, I see this kind of ideas sometimes, and they're usually taken with a lot of skepticism. And I think part of it is because biologists don't have the physics education that you need to kind of even look at this. And I think we're at the point where cross interdisciplinary approaches are going to be like the answer to all this, and it's not quite there yet. So yeah, I think it's a great idea, and I think biologists are not necessarily as open to it as they should be.
Well, I think, for example, a number of years ago, it was discovered how geckos are able to stick on walls. That there are quantum effects with that, right? I think.
And I'm trying to get people to perform some of my quantum mechanical experiments on the grounds that it may have some implications for protein folding times and so forth. And you guys are encouraging me and pushing that.
Well, I think, because this is what I found is true of every other potentially exploitable chemical process, if quantum effects are relevant to biological systems, there will be a virus out there that uses them. And
I quote you on that.
well, I don't know, because I don't think it's been demonstrated. But I would bet that if you really want to demonstrate it, the first place to look is in viruses, because in my experience, every new thing we figure out in biology, within a year, we figure out a virus that exploits that new thing. Always.
You think of computers, it works exactly the same way, I guess, yeah. But viruses only want to propagate. They want to be successful in propagating. They don't care about us or about anything else. They just, and I don't.
One of the striking features of the principal algorithms in artificial intelligence is that all of them are stolen from biology, from evolution. Neural nets, genetic algorithms, and swarm techniques are the three principal algorithms, and they are all stolen. And if there are more out there, the data scientists will be stealing those too.
Yep.
Best engineering ideas come from nature.
We just got we have a 2-minute warning, but apparently we have a little bit of a grace period. I guess there's a benefit to being the final event on the track, and our kindly tech guy is saying, don't sweat it. But just to be respectful of our audience, I'd like to ask that the assembled multitude, that the panelists come in with some some final thoughts about, death to life to death here and so forth. Since you led off, John, would you care to lead off again?
Sure. My final thought is I think that life takes more forms than we're prepared to relate to. I would point to so many episodes of Star Trek: The Next Generation that it's almost a cliche that explored these concepts, and I think that type of fiction is actually a good starting point for asking questions about our natural world. And I guess I'd be remiss not to take the opportunity to shamelessly plug my newsletter again. It's COVID Transmissions, and I'll drop a link in the chat one more time.
Okay. Doug?
I'll say thank you all for this awesome discussion. And now I'm going to thank viruses for two things. One is for their contributions to the development of CRISPR. We didn't really talk about CRISPR, but it's an amazing gene editing technology that was developed in bacteria because of viruses, and shaped by viruses certainly, and then shaped by human hands. And then that was awarded a Nobel Prize in October to Jennifer Doudna and Emmanuel Charpentier for their amazing work on making gene editing a lot easier. I will also thank viruses too, because in my fiction, I write a lot about how gene editing and gene modification can go horribly wrong. And I see that is a possibility for our species in the future. And viruses have also developed and evolved wonderful ways of shutting down the natural CRISPR systems in bacteria. And I'm hopeful that maybe some of those proteins and some of those ways we'll be able to use to help ourselves control this technology. So thank you, viruses. And here's my cat.
Right. Speaking of parasitic organisms,
With a charge.
I remember in Guns, Germs, and Steel, what was the name of the author of that, said that only 12 species have been domesticated on this planet, 11 by us and 1 by cats. Anna, last licks.
So yeah, I just wanted my two cents would be that any organism, so survival and life and death on a big sense matter only when you think of species. So evolution and biology don't think of individuals, they think of species. So humans are the only species that evolved to the point that we can finally start thinking about individuals. And this is, I guess, when we're having all these discussions, because from the coronavirus, for example, point of view, coronavirus wants to propagate, and it's pretty successful, not only in terms of its own propagation, but also in terms like in the human species, it kills off the weakest, but it doesn't kill off the species. So the human species are actually arguably better off if you kill off all the weakest and let the strongest ones thrive. But I mean, I think for the first time in our lifetime and evolution, we can think about individuals and how we can overcome the evolutionary trends of life and death to actually kind of make sure that people survive and that people don't die based on like this very
that's
considerations.
elements of good news from the coronavirus.
Right. And I think it's like, so our advances on the vaccine are really amazing, and it's like
Yeah,
unprecedented and historical. And yeah, I was very excited to hear that. I remember last year, John and I were on the same panel talking about the mRNA vaccine, and I think we're much more skeptical than we are today. But yeah, so I think kind of keeping this perspective is important when we think about those diseases.
That's excellent. Yeah, Pam?
You know, I really appreciated the perspective that the virus itself is inert and not alive, but that the dynamic process is alive, and that I just really appreciated that way of looking at it. And the other thing that occurred to me when we were talking about quarantine and the plague and things like that is this idea that it's not clear to me that there's this concept called sickness behavior, that when your immune system gets riled up, you know, your dopamine receptors get, you know, blocked and things like that. You feel less sociable. So it strikes me that sickness behavior evolved in response to viruses, and I guess I never really thought of it that way. Viruses and other things, you know, it doesn't take that much to elicit it. So, yeah.
James?
Well, it turns out these things were only mostly dead, right?
Sorry, what was that?
No, I just said, it turns out these things were only mostly dead.
Yes. We're coming right back to the title of this panel. Yeah, I am awed by the diversity in life, and I certainly have learned quite a bit actually just from this panel, from prepping for it, and then from you guys, the different perspectives and so forth. So I'd like to thank the panelists for their good work and thank the audience for their attention. I put a couple of useful references into the chat. Does anybody have any final things to put into the chat for people to take away with, or shall we just call this a wrap? Okay, well, that's a wrap, guys, and have a good evening and rest of the con, and thanks very much for your time and work and insights.
Thank you.
Thank you.
This was fantastic. Thanks to everyone.
Thank you for moderating. That was wonderful.
Thank you.
I had a lot of fun with this. I knew it was going to be good.
And I learned a lot from all of you. Thank you so much.
Same.
Same thing. Thank you.
Okay, take care all.
Take care, everyone.
Good night.
Good night.